Hello again. This is the second in a series of 2 presentations on the introduction to the endocrine system. And we had ended the previous presentation looking at insulin as far as an endocrine reflex pathway would be concerned. And now we can consider a simpler one, parathyroid hormone. So the plasma calcium levels are having direct effects on the parathyroid cells of the parathyroid gland. And low plasma calcium levels are going to increase the secretory rate and the synthesis rate of parathyroid hormone. So the local concentration in the interstitial fluid will be increased, washed away into the blood. General systemic levels of parathyroid hormone, or PTH, would be increased and delivered to the target tissue. So the interstitial concentrations would be increased everywhere the blood goes, where there's capillary beds, where there are target cells expressing the receptors on their surface, the bone and the kidney. We will have responses. So there would be increased bone resorption. So remember, low calcium is what brought this about. So we want to add calcium to the blood. So where can we get it from? The bone, by resorbing it, or the kidney to reabsorb it. So it's been filtered. We want to save all that we can. And then the production of calcitriol leads to increased intestinal absorption of calcium. So we try to get as much as we can from the diet, target our reserves in the bone. We don't want to take a lot from there, but we go there first because we know we can get it immediately, right? At the same time, we are sending the signal to the intestine to absorb whatever there is there, and hopefully there's enough in the diet to prevent too much resorption from the bone. Either way, all 3 ways, get it into the blood and then keep it in the blood rather than letting it leave in the urine. And we can bring calcium levels back into the normal range. And when they reach that normal range, they will then, through a negative feedback, turn off the secretory mechanisms at the parathyroid cell. So, in effect, parathyroid hormone increases plasma calcium levels. The parathyroid cells are activated by a low or decreased plasma calcium concentration. Now we can begin to consider the neurohormones as major groups. So the adrenal medulla actually is a collection of cells that are of neural origin, so they can be considered as neural types of cells. So the catecholamines from the adrenal medulla, epinephrine and norepinephrine, and some dopamine gets out too, are considered neurohormones. And then at the hypothalamus, there will be neurohormones that will regulate the anterior pituitary and be released directly at the posterior pituitary. So we'll look at the anatomy of the hypothalamus and the pituitary to understand the distinction between these 2 systems. So here we see a midline view of a young individual's head, and we can see the sphenoid bone here with the pituitary fossa in the sella turcica of the sphenoid bone, the Turkish saddle that it sits in. So this is the pituitary gland. Toward the nose is anterior. Toward the back of the head, where the cerebellum is the posterior. So we zoom in on this squared box here, and we see this pinkish color is the anterior pituitary, and this whitish colored portion is the posterior pituitary. The anatomical distinctions go into much greater detail, but we'll be fine just calling it the anterior and the posterior pituitary. Posterior pituitary is continuous with the neural tissue of the hypothalamus, so it is made up of neural tissue. So as you know, it's also called the neurohypophysis. And the anterior pituitary has glandular cells in it, so it's referred to as the adenohypophysis. those are safe terms for you to use. The neural hypophysis, posterior pituitary, is suspended from the base of the hypothalamus by a stalk called the infundibulum, or the pituitary stalk. And some of the glandular adenohypophysis surrounds the pituitary stalk as well. So we see that is always represented. But we're just going to say anterior pituitary and posterior pituitary to be safe. So the posterior pituitary, we'll see in a series of slides to go through this. There are two hormones, the vasopressin. What was the other name for vasopressin? ADH, which stands for antidiuretic hormone. Very good. Oxytocin is the second. And we talked about oxytocin with the positive feedback system. So we're somewhat familiar with that. So now we want to think about synthesis, transport, and release and delivery to the systemic circulation. So from the posterior pituitary, we know that there are neuron cell bodies of origin, where it's synthesized and packaged, in the hypothalamus, and the axons continue straight through the infundibular stalk, forming the neurohypophyseal tract, a collection of axons or nerve fibers in the central nervous system is a tract. So this is the neurohypophyseal tract, the axons going through the pituitary stalk into the posterior pituitary. So the hormone is made and packaged in the cell body of the neuron within the hypothalamus itself. Then the vesicles are transported down the axon through the and the axons are in the neurohypophyseal tract, hypothalamo-neurohypophyseal tract, to be specific. And they reach the terminal within the neurohypophysis or posterior pituitary. So they are stored there and they're ready for release. Remember, the prohormone is being processed as it's being delivered down to the nerve terminal. And then there is release when the stimulus activates the neuron such that it increases its firing rate. Remember, there's a basal firing rate, maybe one action potential a second, a frequency of 1 hertz, right? And that might go up to 10 hertz under certain situations. It might go up to, 100 hertz during delivery if this was oxytocin, right? Labor and delivery. And so more gets dumped into the interstitial compartment, increased concentration, so you have a greater concentration gradient, so more is washed away through the blood. So the capillary bed here is profuse. It's a rich capillary bed. You want to wash away the hormones. You want the blood to flow through there profusely so that you maintain that concentration gradient to pull the hormone out of the interstitial fluid compartment. So the hormones are released and ultimately get into the blood. Right? So it's not released directly into the blood, goes to the interstitial space first and then into the blood. I am a stickler for that specific. The anterior pituitary gland, now remember we're introducing it here, so as we go through different systems, reproductive and metabolism and stress and so on and so forth, in the reproductive system again, each of these will be looked at in more detail. So this is just to summarize and introduce and to review for you, hopefully. So there are hypothalamic hormones such as prolactin releasing factors or a prolactin inhibiting factor, which is known to be dopamine, that will affect the prolactin release in the anterior pituitary. Thyrotropin releasing hormone will target the cells in the anterior pituitary that synthesize and secrete the TSH, thyroid stimulating hormone, that obviously target the thyroid gland, right? Prolactin is going to promote lactation, so the breast tissue is the target. The thyroid gland is going to synthesize and secrete the thyroid hormones. We saw triiodothyronine and tetraiodothyronine. CRH, corticotropin releasing hormone. Corticotropin is ACTH, adrenocorticotropic hormone. So the CRH stimulates the release of ACTH that targets the adrenal cortex for the cortisol production. Not the aldosterol. The adrenocortical androgens are affected to some degree, but not the aldosterone that we saw before. And there's target cells distributed in many tissues. The growth hormone releasing hormone, or GHRH, also called somatolibrin, like liberate, and somatostatin, right? So somatolibrin, growth hormone releasing hormone, will stimulate or free up the growth hormone. Somatostatin is going to stop it. So this double line is the inhibitory effect. And then the growth hormone will target the liver or many tissues directly, then the liver, and we'll look at this in a little more detail. Then the, the gonadotropin releasing hormone, so notice lowercase n to distinguish between the uppercase G, uppercase H growth hormone. And the two pituitary gonadotropins, as they're called, FSH and LH, follicle-stimulating hormone and luteinizing hormone, target endocrine cells of the gonads, as we had discussed, bringing about androgen synthesis and release in the male, estrogens and progestins in the female, targeting many tissues, as well as germ cells of the gonads, and the gonadotropins themselves contribute to sustaining gamete production. So here we're picturing on the top right the hypothalamo-pituitary axis with neurons in the hypothalamus that are going to release their hormones into the blood. So it's still a long, distant effect of the between the stimulus and the site of release. Then the hypothalamic hormones will be washed through the capillary beds, and we'll give the details of this in a moment, to the anterior pituitary, where it will find its target cells. So in considering the endocrine control of the hypothalamo-pituitary axes, we think of three levels. The hypothalamic stimulation coming from the central nervous system, the pituitary stimulation from the hypothalamic trophic hormones, and the endocrine gland stimulation from the pituitary trophic hormones. So the model system that I will be describing is a 3-gland, 3-hormone system. So gland 1, hypothalamus, with hormones 1, the hypothalamic trophic hormones. Gland 2, the pituitary, with hormones 2 that target the peripheral endocrine glands. And then hormones 3 coming from those. So here's hormones 2, the pituitary trophic hormones. And then hormones 3 are the endocrine gland hormones themselves. So this isn't laid out exactly to show the 3-gland, 3-hormone system. Just let me get the pen. Pointer options. Let me get a felt tip pen. Here it comes. So it would be gland 1, to do it in a column, hormones 1, gland 2, hormones 2, glands 3, and then hormones 3, right? So you'd be able to say this is the pituitary, this is the hormone or hormones. So for the liver it'd be growth hormone releasing hormone and somatostatin, right? So 2 hormones from gland 1. Gland 2 would be the anterior pituitary. Hormone 2 would be growth hormone. Gland 3 would be the liver, I guess. And hormones 3, so Some match this directly, and others do not. And that's why we'll take a look at a couple of them, still in this introductory level. So there's also going to be negative feedback controls. So think of it. The stimulus, hypothalamus, I call it gland 1. They got IC1, IC2, and IC3. And then hormone 1, hormone 2, and hormone 3. And I apologize. I should have looked at the figure legend. You can to see what the the IC is. But for me, I call it gland 1, gland 2. So 3 gland, 3 hormone system, right? So here we have it. So I'm going to say gland 1, the hypothalamus, hormones 1, gland 2, the anterior pituitary, hormones 2, gland 3, the endocrine gland itself, peripheral endocrine gland, and hormones 3. So you're going to have regulation within this. It's always a stimulus with some biological response system and regulatory system, and a response at the other end, right? So the regulatory system will either be a long loop negative feedback from hormones 3 onto gland 2, regulating hormones 2, and onto gland 1, regulating hormones 1. There's a short loop negative feedback, and then some people even talk about an ultra short loop negative feedback. You typically don't hear too much about these, the short loop, but the long loop is the one that we hear about the most. So the control pathway for cortisol secretion is what we can consider in this slide then. And we are seeing the gland 1, gland 2, gland 3. Hormone 1 is the corticotropin-releasing hormone, targeting anterior pituitary cells that synthesize and secrete ACTH, hormone 2, targeting cells of the adrenal cortex that synthesize and release cortisol, and then the cortisol in the blood will have the negative feedback. They're showing it at the hormones, but it's actually targeting the cells that are synthesizing and releasing hormones 1 and hormones 2. Then the target tissue will be where we would find the response to the hormone 3, the cortisol. The hypothalamic hypophyseal, apologize for the dog barking there. Try to snap my fingers and make her stop. That's the feedback you get, right? So the hypothalamo-hypophyseal portal system is what we see in this slide. I said we'd talk about the vascular system. So there's 2 capillary beds for the control of the anterior pituitary, and then there's a 3rd capillary bed in the posterior pituitary. But right now we're thinking about the control of the anterior pituitary, so there's a primary capillary bed. The neurons synthesizing the trophic hormones release them into capillaries in the primary portal system. Primary capillary bed of the portal system. Portal vessels carry the blood from the first capillary bed to the secondary capillary bed. The secondary capillary bed is within the pituitary, the anterior pituitary. So remember, the endocrine glands have a very rich capillary bed. Endocrine tissues, where the endocrine cells are located, like the islets of Langerhans and the pancreas, are going to be highly vascularized as well. The adrenal cortex is highly vascularized, as we see here, and the adrenal medulla, that's an endocrine organ. So wherever you're going to get the release of a hormone, you're going to have a rich capillary bed. So and actually what drew attention to the possibility of the hypothalamus regulating the anterior pituitary was the vascularity of it in the first place. So it was these unique capillary beds joined by blood vessels. They're not veins, because veins are between the arterial side and the heart, for return to the heart. So you typically go from an artery to a capillary to a vein, but here these vessels went to another capillary bed. So it's a portal system. And of course the hepatic portal system had been identified and studied in great detail long before anyone saw this, so they recognized immediately that this is what it was. Anyway, so that describes the anatomy of it. So we see the termination of the gland 1 cells onto the primary portal loops, capillary beds, and then the portal vessels carry it to the target cells, and then, of course, the venous drainage takes it to the heart, through the lungs, into the left ventricle, out of the left ventricle through the aorta to be delivered to the blood, to the target tissues, prolactin to the mammary glands, the milk-producing cells themselves, growth hormone to the musculoskeletal system and to the liver, thyroid stimulating hormone, and remember, the blood is going everywhere from the aorta, right? So all tissues are receiving the blood. All interstitial fluid compartments are experiencing a certain basal or increased or decreased level of these hormones. The target cells in those tissues, in those organs, will be affected. Anyway. As I said, these pictures always make it seem like, oh, it goes right from the pituitary nose to just go straight to the thyroid, but it's going everywhere. It's only the thyroid that's going to affect. ACTH, target cells in the adrenal cortex, and gonadotropins in the gonads, so nothing new there. So we're going to consider a complex endocrine pathway, that for growth hormone, as I mentioned. It's an example of a complex endocrine pathway. And I thought we going to go through the slides. So we see the hypothalamus, gland 1, hormone 1 into the primary capillary loop, growth hormone releasing hormone. Portal capillaries carry it to the anterior pituitary, and it diffuses into the interstitial fluid there. And see, the degradation and removal here is important to sustain a concentration gradient of higher here and lower in the tissue, so it diffuses in. Otherwise, it just washes away and gets diluted into the peripheral plasma. And then the target cells, the GH, growth hormone, or somatotropic cells in the anterior pituitary, bind the GHRH, and it stimulates synthesis and secretion. You just secreted it, so you got to synthesize more to replace it. Into the interstitial fluid, into the venules, back to the heart, right heart, lungs, left heart, aorta, finally to the liver. See, this shouldn't be blue, it should be red, right? It's coming from the aorta back, finally to the systemic distribution, including the liver, and at the liver, binding to hepatocytes, the liver cells, and causing stimulating synthesis and release of insulin-like growth factors, IGFs, and that gets into the systemic circulation. And collectively, they target receptors in tissue in the bone and the soft tissue to contribute to growth. So there would be an animation for that. The hormone interactions is the next idea we want to consider, and we're thinking of synergism. So multiple stimuli can bring about a response that's different than any one of the stimuli alone. So it's more than additive. So you can have stimulus 1 with response A and stimulus 2 with response B. An additive response of stimulus 1 and stimulus 2 would be A plus B. But what if your response was greater than A plus B? So that's a synergism. So it's more than just the results of the direct results of the two stimuli added together. There's a permissiveness where we need a second hormone to get the full expression of another hormone response, and an antagonism like glucagon opposing insulin. So here's a graph that shows an example of synergism. And on the x-axis we have time in hours. You can read the textbook to get more information about this. I'm only going to point out a couple of things because I like this one for exam purposes. So we have time in hours on the x-axis. Always look at not just the what is being measured, but the units. And then the blood glucose in milligrams per deciliter. So how freely can you convert this to other units, milliliters and stuff like that. So we see that blood glucose is increased by cortisol, not dramatically, but here is 100 milligrams per deciliter. That's upper end of normal, right? The last couple times I've gone in, my resting morning blood glucose is 100, and doctor doesn't really like that. He'd like to see it to be more like an 80, right? So let's get it into perspective, and that's milligrams per deciliter. So 150, 200, 250 is really high. So there's an extreme situation when you're going to pump it up that high. So glucagon, epinephrine, and cortisol all together will produce this really dramatic, very high change in the plasma levels over a period of 5 hours. Glucagon and epinephrine will only bring it up roughly, around 130, 150. Epinephrine alone will only bring it to about 125. Glucagon won't have a persistent dramatic effect. So you can see that glucagon and epinephrine, if you add them together, would only come up somewhere in here. So you have is this level greater than these two added together? So you take this 100 and add it to this 100 and whatever. Add those two together. Do you get this? And if it exceeds that, then it is synergism. So there can be endocrine pathologies as well. So here we see exogenous cortisol being added. Some people are given this. Remember, there's non-steroidal anti-inflammatories. Well, the steroidal anti-inflammatories are exogenous cortisol. Right? So for various reasons, people are given steroids, not the androgenic steroids, but just anti-inflammatory steroids. So the exogenous, just as we talked about with the exogenous androgenic steroids acting like testosterone, they're going to have a negative feedback at the level of the hypothalamus and the pituitary, decreasing synthesis and secretion of those two. Therefore, the endogenous cortisol levels are going to decrease. So in an individual that was taking exogenous androgenic steroids, their endogenous normal testosterone levels would be very low. Right? So their own testosterone, which can be detected and measured, would be very low. So that's one indicator of steroid use. Low endogenous cortisol, if it could be measured, depends upon the chemical formula of the exogenous steroid that's being given. Whether your instrument that you'd be using to measure it in the blood could distinguish between them, right? If they're exactly the same, you wouldn't be able to say, well, this is what he swallowed and this is what came from the adrenal gland, right? But anyway, for the anabolic steroids, androgenic steroids, they are distinct molecules and they can be measured separately. Anyway, if you can distinguish, then you can tell that the endogenous is reduced. And then animal studies have shown this extensively. So the effect on the target tissue is the response is controlled entirely by the exogenous cortisol. So just recently, my son hurt his ankle and it swelled up. So the emergency room, they gave him methylprednisone. And the dose for the first day was 6 of them, 4 milligrams each. And he took them throughout the first day. The next day there was one less. The next day the two less. The next day three less. Ultimately until it got down to just one pill in one day, and then he stopped. So why was that? You get hit with a first dose to start with to have the anti-inflammatory benefit at the swollen ankle that was just sprained. So it's going to shut down all this stuff. And so you slowly have to bleed or wean off the high dose, and any dose would be beneficial, but you want a large dose to start with to get a maximum anti-inflammatory benefit at the site of injury, and then slowly come off so that the plasma levels of cortisol will be closer to the normal range as a balance between the recovering endogenous and decreased exogenous doses. You can't just come off a high cortisol, high methylprednisone dose immediately. You have to taper to allow the endogenous system to recover. So as I said, in short term, the endogenous system recovers. If long-term abuse of illegal anabolic steroids, those acting like the androgens, the hypothalamic-pituitary gonadal system sometimes never recovers. So there has to be a lifelong supplement of exogenous testosterone just to keep it in normal range levels for normal life functions. So endocrine pathologies can be in a couple categories: hypersecretion with excesses, tumors or cancers, grave disease with the thyroxine being affected. The hyposecretion, a deficient hormone, so goiter where thyroxine again is affected, and diabetes where insulin is affected. So the pathologies also can be related to abnormal receptors where they're downregulated, such as in hyperinsulinemia. So you're not getting the cellular response to act for the skeletal muscle and the liver to act like sponges, glucose sponges, and pull the glucose out of the blood. So the pancreas is experiencing this really high level of glucose and just keeps pumping out insulin, right? So you've downregulated the receptors at the liver and the skeletal muscle. The pancreas is just saying, here's more insulin. Get rid of the glucose. Transduction abnormalities such as testicular feminization syndrome or pseudo-hypothyroidism, and abnormalities of control mechanisms. So quite obviously I'm trying to streamline these recorded presentations as much as possible. You're referred to the textbook for the additional information, descriptions on these cases, unless we encounter them in these upcoming slides. So primary and secondary pathologies will be considered next. Just was checking to see if we had a sequential series of slides. In panel A, we see secondary hypersecretion due to a hypothalamic problem. So if there's a pathology in the hypothalamus causing an increase in CRH, then there would be an increased ACTH and an increased cortisol, and the negative feedback fails. So there's clinical tests where you can administer synthetic cortisol and measure the plasma ACTH levels. So there are ways of investigating clinically what would be observed. So in a hypothalamic level with an increased trophic hormone, you'd see high levels of all three. Now these hypothalamic hormones, by the time they get into the systemic circulation blood, they're very low levels, so not all of them can be measured. CRH is where you can pick up some plasma levels, but you've got to have a really good assay with a high sensitivity and specificity because the levels are so low. But ACTH is easily measured and cortisol is easily measured. A secondary hypersecretion due to a pituitary problem. So in this case, the ACTH is going to be high, and the cortisol is going to be high. So hormones 2 and 3 are going to be high, but hormone 1 is going to be shut down from negative feedback. And then a primary hypersecretion due to problem with the adrenal cortex. Well, then hormone 3 is going to be high, and hormones 2 and 1 are going to be shut down from negative feedback, right? So again, if you have access to the animation, this would be a good time to watch that, to study the stress response. So let's consider a pathology of hypocortisolism. So a hyposecretion from damage to the pituitary, or hyposecretion from atrophy of the adrenal cortex. So the challenge here is to fill in what would be happening in each of these. So let's switch my pen. There's the pointer. Get my felt tip pen again. So we're talking about a hyposecretion from damage to the pituitary. So the CRH is going to be down, right? And what about ACTH? It's not going to have its trophic stimulus, so ACTH is going to be down. If ACTH is down, then cortisol is down. So then there would be the symptoms of deficiency. A hyposecretion from atrophy of the adrenal cortex, so cortisol would be down, and if this level is low, then ACTH and CRH are going to be increased. Right? So always remember the negative feedback at the two levels. That one continues up there, right? Right? So if you lose the adrenal cortex, you don't have any negative feedback, these are going to go up. Now with a decrease in the hormones 1, you still would have the negative feedback. I don't know why it jumps like that. Sorry about it. So you would think that the decreased cortisol would still remove negative feedback at the anterior pituitary, which isn't damaged. It's obviously not going to be functioning at the hypothalamus because you got damage. Oh, it's damage to the pituitary. Sorry. Yeah, so it would be cut off here too because you've got a decrease in the ACTH. Ooh, so the hypothalamus is still functional. Sorry about that. So the first one we did was if it was a hypothalamic damage. Ooh, that's weird. Anyway, let's correct it then. Hopefully you picked up on that. I apologize. So if the pituitary was damaged so that you're not getting ACTH, then the negative feedback to the hypothalamus would still be intact and hormone 1 would be up. But it can't do anything at the pituitary. ACTH can't be stimulated, can't be produced and released. So cortisol's still going to be down. But if cortisol is down, hormone 1 is going to go up. Hormone 2, if everything was working, would go up too, right? As we saw over here. So in other words, over here on the left, once I corrected it, for pituitary damage, the first one we did, as you recall, was hypothalamic damage. For pituitary damage, you take this out of the picture, remove the pituitary, and you've got the same appearance. Okay. Please do not let me have confused you on that. So think it through and stick with it. Okay, so hormone evolution. There's evolutionary conservation of hormone function across many species. It's really kind of cool to see how it works. Obviously, a clam doesn't have kidneys, so antidiuretic hormone isn't quite doing this, acting on the same organ in those types of organisms. But still, as far as fluid homeostasis, it plays a significant role. So the comparative endocrinology becomes important in that. The pineal gland and melatonin is an example. So it influences the body clock and has antioxidant activity. Other roles still need research as far as seasonal affective disorder. What's the other? Right? And sexual behavior. I think apologize. Should have double-checked on that. So the pineal gland and melatonin are shown in this slide. So we see again the midline section of a brain inside this individual's head. And you can see the pink pineal gland here between the cerebellum and the cerebrum. So the hypothalamus and the pituitary were here, as you recall. So above the hypothalamus is the thalamus. Above the thalamus is the epithalamus, and there's the pineal gland. So the pineal gland is responsible for the synthesis and release of melatonin. It freely passes into the plasma as it's synthesized. It's from a modified amino acid, tryptophan. Tryptophan is also used to make serotonin. So serotonin and melatonin are interconvertible depending upon the enzymes. So at night, the enzyme that is responsible for producing the melatonin is high, and during the day it is low, and therefore serotonin levels would be the opposite of this. Serotonin levels would be high during the day and low at night because it's being converted to melatonin. So the tryptophan is used to make the serotonin, and the serotonin is converted to the melatonin. So we see that in humans, the melatonin levels are high at night and are associated with sleep. In nocturnal animals, melatonin levels are still high at night, but they're associated with wakefulness. So it serves as a cue, not as a direct physiological stimulus to bring about a response. So in other words, if you take melatonin, it's not going to put you to sleep. It is associated with somnolescence or sleepiness in humans. Therefore, it does have a somnolescent effect, but more so it contributes to regularity of the cycle and helps you to stick to a stricter sleep schedule. So here we see the pineal gland and melatonin, the melatonin itself. So the tryptophan, so remember the tyrosine had only one 6-membered ring. The cholesterol had rings A, B, C, and D was the pentagon, but the tip of the roof pointed up. So tryptophan has the house upside down with the nitrogen, the amino group in that 5-membered ring. And there's no iodine sticking off it. It's not 2 of the 6-membered rings stuck together with an oxygen. So you should be able to distinguish this from the catecholamines and thyroid hormones and the sterols very easily. And I would have to have a picture of serotonin here. So 5-hydroxytryptamine and melatonin, 5-hydroxy. 1, 2, 3, 4, 5. So that might be the difference there, that the melatonin is methylated. I'm just guessing. Okay, in summary then, we saw the introduction to hormones in this set of lectures. We talked about classifications and features of the hormones. We considered regulation controlled by the endocrine and the nervous systems. We considered interactions of hormones with other hormones and some endocrine pathologies to reinforce the ideas of regulation. And we talked a little bit about hormone evolution, but not in enough detail to think that we would be an expert.